будемо розпочинати зараз вже конференцію секцію молодих вчених. Я хочу подякувати всім доповідачам, всім присутнім за такий жвавий інтерес, за такі цікаві доповіді. Я сподіваюся все ж таки, що ми не останній раз зустрілися і що ми продовжимо з вами співпрацювати. Якщо у вас буде таке бажання якось відкоментувати нашу конференцію, будь ласка, ми чекаємо фідбеку від вас на усіх наших соціальних мережах, у Фейсбуці, в Інстаграмах, можливо, на почту кафедральну, якщо у вас є бажання висловити свою думку щодо організації та проведення нашої конференції. Тому я передаю зараз права модератора Соловйову Аліни Володимирівні, яка буде модератором саме секції молодих вчених, і запрошуємо молодих вчених до плідної співпраці. Будь ласка, я представлю першого доповідача, тому що це якраз і є Соловйова Аліна Володимирівна, аспірант, асистент нашої кафедри біотехнології Національного фармацевтичного університету з доповіддю, присвяченою розробці складу крему для застосування у дерматології з пробіотичними культурами. Будь ласка.
Good afternoon, dear participants. Let me present our work with Olga Kalyuzhnaya, my scientific supervisor. The purpose of our work is to study medicine with probiotic live cells and probiotic metabolizers in order to find promising substances for further development of composition and technology of an effective local acne treatment and prophylactic medicine. So, why are probiotic microbes the future of skincare? Probiotic can shift the skin ecology to favor a diverse and harmonious ecosystem that is healthy enough to resist attacks from pathogens. Most people associate probiotics with maintaining a healthy gut. The pie chart shows that your skin holds a large part of your microbial diversity. Microbes are about 10 times smaller than human epithelial cells, and they are everywhere. So unless you know you are looking for, it's difficult to see the big pictures without the microscope. What do your microbes do for you? You have distinct microbes in your gut, your skin, your oral cavity, your airways, and if you are female, your vagina. In all these sites, your microbes protect you from invalid pathogenic microbes by competitive exclusion. This means that they occupy all the space and consume all the available food that invaders can't colonize. What do probiotics do for your skin? Probiotics protect your skin from pathogens. They can form a layer and use all the available nutrients so that opportunistic pathogens can't colonize. They also produce antimicrobial peptides that make life really through for incoming bad microbes. Probiotic bacteria produce hyaluronic acid, which is extensively moisturizing, active, and naturally hold the moisture on your skin. Many probiotics produce lactic acid and other acids that keeps your skin pH in the optimal range. Probiotic bacteria on the skin can communicate with your skin's immune system to keep it in check so that it doesn't overreact. The improvement of barrier function and also prevent many toxins from penetrating the skin in the first place. Probiotic helps slow the sign of aging by protecting your skin and by keeping your own immune response at bay. They have also been shown to reduce wrinkles and improve skin firmness and elasticity. The skin microbiome runs deep. Before, it was assumed that the skin microbiome was on the surface of the skin and that the tissue below the surface consisted only of human cells. But it was shown that there are a strong population of microbes throughout the epidermis, below the basal membrane, and all the way into the adipose layer below the dermis. The skin microbiome varies between body sites. From a microbe's perspective, the skin can be broadly broken into 3 habitats: dry, moist, and oil. Beneficial microbes use all available nutrients and occupy all the space to exclude opportunistic and pathogenic microbes. Probiotics communicate with human cells to increase their production of antimicrobial peptides, which selectively kills pathogenic microbes but don't harm beneficial microbes. Probiotic microbes produce moisturizing factor like hyaluronic acid. Probiotic microbes communicate with cells in a stratum granulosum to increase the number and quality of tight junction. Tight junction keep water in skin and keep toxin and invalid cells out. There are 4 levels of probiotic in skincare. That is broth, lysate, tindalization, and live probiotic. It is true probiotic product. As part of our work of development of composition and technology of therapeutic and prophylactic medicine with probiotic for treating dermatological problems, were selected 6 samples with a probiotic form from different manufacturers and diverse price segments to determine the probiotic content in them. We were selected these 6 samples, which you can see on the slides. To determine the probiotic content in the samples, we prepared 2 solutions of the sample in polysorbate and saline. Next, we prepared a series of dilution for each sample and so in petri dishes with nutrient medium. Cultivation was carried out under optimal condition for 48 hours. Summarized results of our study showed a large number of colonies for samples BioFresh yogurt of Bulgaria, Elise Cosmetic, and sensitive serum by ESSE. The number of microorganisms for Valmon and healthy skin were much lower, and we saw complete lack of growth for hydropeptide. In the future, we'll all look after the microbes on our skin. We'll stop using antibacterial products that kill both good and bad microbes. We will choose products without synthetic ingredients and harsh preservatives that pollute and destabilize our skin microbiome. The future is probiotic, but not antibiotic, and that future starts now. Thank you for your attention. If anyone have some question, don't hesitate to ask. You can use a chat or switch on your microphone. Так, we have a question from a chat. Доброго дня, Аліна Володимирівна. Наскільки я розумію, в Україні немає лікувального дерматологічного засобу з пробіотиком. Так, доброго дня, Уляна. Дякуємо за запитання. Дійсно, в Україні такого лікувального засобу немає, тому ми з Ольгою Сергіївною обрали це як тему нашого наукового пошуку, щоб обрати раціональний склад та композицію засобу, ефективного засобу, щоб домогтися виживання пробіотичних бактерій протягом сроку зберігання. Є ще запитання? If anyone has questions? Okay, so next speaker is Savkov Ivan Alexeyevich, a PhD student, chair of Industrial Technology of Medicine with course of FCE and SR, Master of Pharmaceutical Science from Vitebsk State Medical University, Republic of Belarus, with report Technological Properties of Red Raspberry Leaves Dry Extracts. Welcome, Ivan. You can start. Ivan, we don't hear you. Ivan, please turn on your microphone.
It's okay?
Yeah, it's okay, thank you.
Good afternoon, dear listeners. Let me present a report on the topic Technological Properties of Red Raspberry Leaves Dry Extract. To select a technology for obtaining tablets of dry extract of red raspberry leaves, it is necessary to study its technological properties. The following technological properties of dry extract of red raspberry leaves were studied: bulk density, tap density, compressibility, and flowability. The bulk density was determined as the ratio of mass of unconsolidated red raspberry leaves dry extract mass to its volume, including the contribution of free space between the particles. The volume density of the red raspberry leaves dry extract was calculated in kilograms per cubic meter using the formula m divided v0. The tap density was determined at the bulk density obtained after mechanical shaking of the reservoir with the powder of dry extract of raspberry leaves. To determine the tap density of 10, 500, and 1250 impacts, the corresponding volume were recorded V10, V500, and V1250. The tap density of the red raspberry leaves dry extract was calculated using the formula m divided v1, where v1 is the final volume of the compacted powder. Flowability is a highly informative value. Knowing the flowability, it is possible to control the technological process without knowing other parameters of the material. Compressibility is the strain of the tablet after release of pressure. The better the compressibility, the higher the tablet hardness is. If the compressibility is low, the tablet will be fragile. Determination of the flowability of dry extract of red raspberry leaves was carried out according to the following method. A sample of powder 25.0 gram was poured into a dry, clean steel funnel with a cone angle of 60 degrees. The funnel hole diameter is 8 millimeters. The funnel was installed on a vibrating machine. After 20 seconds of vibration to obtain stable results, the hole of the funnel was opened and the time during which all powder flow out was noted. 5 determinations were carried out and the average values were calculated. To determine the compressibility, 0.3 gram of powder was compressed into a tablet 9 millimeter in diameter at a pressure of 120 megapascal. The crushing load was determined using an Erveka TBH 100 device. The compressibility of the powder was determined in newtons. The investigated dry extract of red raspberry leaves, in accordance with the classification of flowability of powders, belongs to the class 3, and it's characterized by satisfactory flowability and medium compressibility. The bulk density of the dry extract of red raspberry leaves was 800 kilograms per cubic meter, and the tap density was 1000 kilograms per cubic meter. Therefore, it belongs to the class of medium powders. Thus, based on the studied technological properties of dry extract of red raspberry leaves, to obtain tablets on its basis, direct compression can be proposed with the use of auxiliary substances that improve the flowability of the tablet mass and ensure its volumetric dosage, as well as the granulation operation in the technological stage preparation of tablet mass. Thank you for your attention.
Thank you very much, Ivan Alexeyevich, for such an interesting and full of information report. Does anyone has questions for a speaker? Any questions? Okay, Ivan, I have one. Can you tell us where is more bioactive ingredients in raspberry, in leaves? I mean, in leaves or in fruits?
If we tell about antipyretic activity, more antipyretic components we can find in leaves of raspberry.
Okay, thank you very much. Any questions? Okay, so Ivan, please thank you. Turn off your screen sharing. Ivan, thank you. The next speaker is Shmaida Adam, doctoral candidate from Lodz University of Technology, Faculty of Biotechnology and Food Sciences, Institute of Fermentation Technology and Microbiology, Lodz, Poland, with reports on the techniques in your fridge.
Greetings to everyone.
Oh. Yeah, thank you.
Thank you. Is there a presentation is visible for you all?
Yeah.
Okay, greetings to everyone. Today I would like to talk about ohmic techniques in your fridge. Actually, in the study about meat quality and how ohmic technique can impact in the nearly future this type of study. So let's bring it on. The meat production in the world and in Europe is increasing since probably since ever, and there is the free type of meat that are eat in the world and in the Europe. It's pig meat, beef, and poultry. Poultry become more and more important in animal protein as protein animal protein source. But it's this type of protein source need a lot amount of ground water and other source to produce of this type of protein source. So every waste and every spoilage of such product is a great mess. And what to make it worse, it is estimated even 20% of meat do not reach consumer plate. So extrapolating such information, every 1 in 5 chicken could be killed for nothing. To prevent such a mess, we should develop the method for prevent spoilage in early stage to achieve enough quality in meat product. For now, to achieve it, there is several technique, microbiological technique and physical chemical technique to assess meat quality. In numerous studies, the such parameter like mesophyll, aerobic count, lactic acid bacteria count, Brocotrix, Enterobacteriaceae, and Pseudomonas count are used in terms of physical chemical tests. Texture, color, odor, flavor, and deep losses are assessed in context of food of meat quality, as well as pH and thiobarbituric acid amount. It's a parameter about carboxylic as acid oxidation. So this one are typical used for assess of the quality. But the alternative for that test might be use of ohmic technique, because the quality of meat and for other product is the result of the whole history of product of production to the plate of the consumer. So all of the aspect like animal life, the environment processes, the processing, and packaging, storage condition affect the bacteria profile in product, and as a result, the metabolic pathways occurring in a in given matrix. For example, our chicken meat. So how we can assess what's going on in our product? We can use omic techniques such like genomic transcriptomic to assess the proportion to assess profile of microorganisms present in our product. It's say about how which metabolic pathways are available as result of microbial spoilage, being one of the most important during quality loss of meat product. To assess such profile, there is mainly use of sequencing of environmental genes in meat product, especially use of 16S RNA genes, and as a result we get the profile of bacteria in product. But there we can use also proteomic technique and assess them through nuclear magnetic resonance technique, electromigrative technique, and immuno-based analysis like Western blotting. What is also very important in the point of view of metabolic process occurring in given matrix, we can study metabolomic change in product. For example, using also chromatographic technique, especially interesting in aspects of voltage of metabolite product, are gas chromatography and such like technique, also electronic nose use. And in my opinion, there's two omic technique, including genomics, metagenomics, and metabolomics, are most important for quality assessment of meat product. Here I show you the quick review for several study about study of profile microbiome of meat product. So how I said previously, there is mainly 16S RNA sequencing use for such a study. And maybe what are main conclusion of such a study, because there is some general conclusion we can get out from this study. The main from all of it is that processing technique, patching condition highly affect on product we occur in the meat. The diversity of microorganisms, of profile of microorganisms present in product is dropping during processing and during storage, because of development of some specific group of organisms, specific group that result in spoilage, actually, such as Pseudomonas species, and when they develop, they actually decrease overall diversity of the product. They in their study have found some specific group of microorganisms that can be used to assess risk of early spoilage during further product life. How it was say in Handley et al. They found that Pseudomonas enterobacteriaceae group Chrysobacterium might be such an indicator organisms. But how about the metabolomic technique and the detection of metabolite in product? Here, the main techniques used are gas chromatography coupled with mass spectrometry, and often to connect with solid phase microextraction, which increase detectability of individual compounds. So here I show you there's also the metabolite profile that is highly correlate with individual aspects of meat quality. So the change of metabolite profile appearing of some compounds and decreasing in the signal of other compounds might indicate the loss in the product quality. But the use of gas chromatography is, in my opinion, not so useful for assess of quality in technical scale. But there are other techniques that can allow for such assessment. And there is also use of metal oxide electrode compound in specific devices such as electronic nose that could be able to indicate the quality degradation in product, in meat product, in a large scale. Such devices can be used in the nearly future for technical scale assessment of quality. But backing to chromatographic analysis, because electronic nose do not show the information about metabolic profile, but in chromatographic coupled with mass spectrometry, we achieve such information, and such information can be also used for determination of some individual organism presence in given matrix. For example, the Caratio et al. Showed that isoamyl chloride, isovalerianic acid in chicken meat matrix can indicate presence of Salmonella typhimurium cerevisiae in product, and such a profile is changed between given organism, pathogenic organisms, and given food, given meat product matrix. So I hope that in near future there's gonna be some kind of algorithm that allow us for assessment of product safety of individual product, because in general, use of omic technique increase the detectionability of different pathogens. We actually don't have to know exactly which pathogen we're tracking, and how we see on in this table, there is still a large amount of unknown or unspecified outbreaks or epidemic events without no agent we should track. So to keep it together, the conclusion is that shelf life and quality and meat safety, and therefore product safety, largely depend on the activity of inhabiting bacteria profile. And all of the condition that affect product during processing and to the end product affect on this profile, and this profile affect metabolite processes during storage, and resulting in metabolite profile, which can be used for further evaluation of quality and safety. The most important technique used for now in meat quality study are sequencing of 16 sRNA gene fragments, and to assess the quality, the metabolites profile. The most often used technique is gas chromatography coupled with mass spectrometry. And I hope that in nearly future, such technique increase the tracking of meat quality in commercial institutions. I think that the most important of omic technique use in meat quality assessment. Thank you for your attention. If there's any question to this presentation, I will be happy to answer it. Thank you.
Thank you, Adam, very much for such an important research. We have a question from a chat. Question from Uliana Starushenko. As far as I understand, your work is a review of omic approaches in the studies of spoilage phenomenon. Are these methods used in practice to determine the spoilage of meats in industrial plants or industrial laboratories in Poland?
Actually, there is no use of in it in a term of commercial assessment, but there are laboratory study in this field with cooperation with commercial with industry. Actually, we conduct such a experiments, and I can confirm that the presence of given compounds such like acetylene increasing signal is related to spalling phenomenon with positive correlation.
Thank you very much. Thank you. Any questions for Adam? I don't see. So next speaker is Tederso Triin, PhD student from Estonian University of Life Sciences, Institute of Veterinary Medicine and Animal Sciences, Chair of Food Hygiene and Veterinary Public Health, Tartu, Estonia, with the report The Prevalence and Antimicrobial Resistance of Campylobacter isolated from fresh broiler chicken meat at Estonian retail level.
Thank you. Can you hear me?
Yes.
Yes. Okay. Yes, welcome to all. And I'm going to speak about the prevalence and antimicrobial resistance of Campylobacter isolated from fresh broiler chicken meat sold at the Estonian retail level. So the main reason why our research group focuses on Campylobacter is because Campylobacter continues to be the most common cause of foodborne bacterial gastroenteritis in both in EU and as well as in Estonia. You can see here that the average in EU is 240,000 cases per year. And in Estonia, according to the Estonian Health Board, it's around 350 cases, Campylobacter enteritis cases per year. So it's quite a big problem because the number of cases is even higher than the number of salmonellosis or yersiniosis or listeriosis. So the aim of our study was to investigate, yes, the prevalence, counts, and the antimicrobial resistance of Campylobacter isolated from fresh broiler chicken meat of Estonian, Latvian, and Lithuanian origin collected at Estonian retail outlets. While we are primarily interested in fresh chicken broiler meat, that's because poultry meat, especially fresh chicken broiler meat, continues to be frequently contaminated with Campylobacter and is the most likely cause of Campylobacter enteritis in humans. So if poultry meat is contaminated with Campylobacter at the retail level, then Campylobacters are more likely to spread into the food handling, preparation, and consumption chain, so posing a risk to human health. And according to the EFSA, European Food Safety Authority, the highest risk to humans, human health from Campylobacter is more than 1,000 colony-forming units per grams of product. So the presence of Campylobacter in poultry meat indicates a wider problem, and it's likely that these bacteria have spread throughout the poultry production chain, from the farm level to the slaughterhouse, through the slaughterhouse level and to the retail level. So between September 2018 to August 2019, we examined a total of 399 samples of fresh broiler chicken meat. We collected products originating from Estonia, Latvia, and Lithuania that were sold at the Estonian retail level, and we used the microbiological analyses that were based on ISO standards. The first method was that we performed was the detection method to get to know if the product is contaminated with Campylobacter, and the other one was the colony count technique to get to know how big are the numbers in a certain product. So in 2018 to 2019, we examined a total of 399 samples, and 35.3% of samples turned to be positive or contaminated with Campylobacter. And we examined a total of 133 samples of Estonian origin, and we managed to identify Campylobacter positive samples only from 2 products. So it means that the Campylobacter, it was 1.5% of products were positive. So, and but we, yes, here. But if you look at the Latvian and Lithuanian products, you can see that Latvian products, the positive number of samples were 15, and for Lithuania it was 89 from 133 samples turned to be positive. So Campylobacter is present more in the summer and autumn months, but in the winter and spring months, there is usually no or very low contamination. And this can be explained by the heat and moist content of Campylobacter. And during this, our study period, the contamination of Campylobacter appeared in samples of Estonian origin only in July, when we managed to identify 2 positive samples. And in the remaining months, we were not able to isolate Campylobacter. But for products of Latvian origin, we saw a clear seasonal variability that's shown in the yellow. And but poultry meat samples of Lithuanian origin, we saw high prevalence rates throughout the study period, throughout the year. So it was ranging from 28.6 to 100%, and including in the winter months, where it was 18 to 85.7%. So in addition to the prevalence of Campylobacter, it's very important to assess also the number of the bacteria in the product, which determines the risk to humans and to human health from the consumption of potentially contaminated poultry meat. So this following slide gives you an overview of the number of Campylobacter numbers in samples. The first 3 columns in the left, you can see these samples that turned out to be negative, where we couldn't detect any Campylobacter. But and also I can say that almost 99% of the samples of Estonian origin were free of Campylobacter. But for Latvia it was 62.4%, and for Lithuania it was 33.1%. And the next column below 100 grams of bacteria show that those samples where the counting method gave us a negative result, but the enrichment method gave us a positive result. And in the case of Estonia, 2 positive samples that continued very low numbers of Campylobacter, less than 100 bacteria per grams. And in the case of Latvia, we saw that almost the overall prevalence was 37%. In 30% of cases, the abundance in the product remained very low, below 100 bacteria per grams. But if you look at the last 3 columns that we were most interested of, you can see these products that had Campylobacter counts more than 1,000 colony forming units per grams. And you can see that 20.3% of Lithuanian origin of samples had the Campylobacter counts more than 1,000 bacteria per grams. So, and in the case of Latvia, it was 0.8. So here you can see which months between the study period Campylobacter was present more than 1,000 colony formed units per grams. And while in poultry meat of Latvian origin, the presence of Campylobacter was more than 1,000 colony forming per grams in only 1 product in February. There was 1,500 CFU per grams, but in the case of Lithuanian products, it was from October 2018 till August 2019. So this slide shows the Lithuanian samples with the Campylobacter counts above 1,000 ceph/grams, and 37% of the samples being between 1,000 to 2,000 ceph/grams, 26% of samples were between 2,000 to 3,000 ceph/grams. But the last chart on the right, you can see that there were 7% of samples that had Campylobacter species more than 5,000 colony forming units per grams. It's quite high. So we also performed the stability of the Campylobacter isolates, and we had in total of 61 isolates. We included also the human strains or human isolates from human Campylobacter enteritis cases, and we had samples from Estonia, 4 samples, Latvia 16, and Lithuania 26 of isolates, and from humans 15 isolates. A total of 46 of the isolates tested were Campylobacter jejuni, and 15 were Campylobacter coli. Campylobacter resistance was higher in nalidixic acid. It was 19.2% of strains, and ciprofloxacin also 90.2%. That were followed by tetracycline. It was 57.4%, streptomycin 42.6%, and erythromycin 6.6%. And all the strains were gentamicin susceptible or sensitive to gentamicin. So the resistance phenotypes of Campylobacter isolates are present here. You can see that 29.5% of the isolates of Campylobacter isolates were resistant to the combination of ciprofloxacin, nalidixic acid, and tetracycline. 23.1% of isolates were resistant to the combination of ciprofloxacin, nalidixic acid, tetracycline, and streptomycin, and 40.6% of the isolates were resistant to ciprofloxacin, nalidixic acid, and streptomycin. So what to conclude? Yeah, the fresh broiled chicken meat originating from Latvia and Lithuania that are sold at the Estonian retail level is often contaminated with Campylobacter during the study period that we see. And the concentration of Campylobacter in products of Latvian origin is mostly low, but the fresh chicken meat products of Lithuanian origin are characterized by high prevalence and high concentration of Campylobacter throughout the study period. And the seasonal peak of Campylobacter contamination is mainly in the warm summer months, but positive samples can still be detected also in other months, especially for the Lithuanian chicken broiler meat sold at the Estonian retail level. And Campylobacter is generally very seldomly detected in fresh broiler chicken meat of Estonian origin. And while Campylobacter isolates of Estonian origin chicken meat proved to be sensitive to tested antibiotics, the isolates of Latvia and Lithuania origin chicken meat were 100% resistant to one or more antibiotic, and 86.7% of the human strains were resistant to one or more antibiotics also. In order to keep I think the Campylobacteriosis as low as possible, more attention has to be given to highlight the contamination in imported broiler chicken products. The cross-contamination needs to be prevented at all levels of the food chain, from the farm to the retail, and effective heat treatment, the core temperature higher than 75 degrees of a poultry meat must be ensured. And I thank you. That was all for me.
Thank
If you have any questions.
It's very interesting report. Any questions for Trine?
Actually, if I can have one question.
Yes.
Very good presentation, Trine.
Thank you.
And I have a question about Salmonella and Campylobacter, because
Yes.
Campylobacter is the most often found to be etiological agent for foodborne diseases. Salmonella is the second. But in the case of epidemiological events, the Salmonella is first one. And how do you think, where is this proportion between the result in illness of Salmonella and Campylobacter, and that in case of epidemiological events?
I think that there could be also more cases of Campylobacteriosis, especially the Campylobacter enteritis cases, because I think just it's self-limiting, the disease, and the people don't go to doctors, so there can be the number of Campylobacteriosis could be even higher than according to EFSA or the ECDC, as we saw. Yeah, but it's more self-limiting. Yeah, but it's still, it depends of the contamination rates as well, how and the immune system of the humans who get the disease or get the bacteria. And it could be a serious disease as well, because there are complications also.
Right. So if I understand it correctly, it is probably that the epidemiological event connected with Campylobacter is even higher than Salmonella, but it's more self-limiting, and that's why it is harder to track back the Campylobacter events. Yeah. Thank you for your answer.
Please.
Thank you. Any questions? Okay, so next speaker is Olga Gnatyuk, student in pharmacy of National University of Pharmacy, Kharkiv, Ukraine, with her report, obtaining an analysis of extract from the fruiting body of the Phallus impudicus mushroom.
Good afternoon.
Good afternoon, Olga.
Okay. The topic of my presentation is obtaining an analysis of extract from the food body, the Phallus impudicus mushroom. Today, the efforts of many scientists from different countries are aimed at finding new drugs that are effective in cancerous disease. In spite of the huge investment they are made to address this problem, the number of cancer patients is steadily increasing. The last 50 years in the study of mycologists, biotechnologists, physicians, more and more attention is paid to macromycetes as potential producers of biological active substances. The most widely mushrooms are used for medical purposes in the country of East Asia, where today 272 types of fungi are used, and other 200 species are studied as permission for the treatment of various diseases, viral, infection, oncological, cardiovascular. In the world of medicine, mushrooms with the proven therapeutic effects such as shiitake, maitake, reishi, cordyceps, coriolus are used. In Ukraine, as effective medicine for oncological disease are Phallus impudicus, Pleurotus usteratus, Formulina veluticus. The anti-tumor properties of skinhorn are associated with polysaccharides found in higher fungi beta-glucans, which activate specific cellular immunity, activate and inhibit the toxic T lymphocytes or natural killer that begin to produce performing protein, which ruin cancer cells. Science also found volatile substances, fungal phytocytes, which have a detrimental effect on virus, ranging from rhinovirus to AIDS and other. The champions of this amount of this volatile are the Japanese shiitake mushroom and our common stinkhorn. Aim of the study is carrying out phytochemical studies of skinhorn fungus tincture. The mushroom was harvested by us in July 2018. In the work used freshly picked and dry raw materials. Drying of the fruit body of the mushroom was carried out in cool condition at a temperature of 2 to 8 degrees Celsius. As at thermal drying, the fungus entered the stage of maturity, intensive growth, in which the mushroom, according to the literature, is conditional edible. Since mushroom tincture are prepared in traditional medicine using vodka, it was interesting to study how ethanol strange affect the extraction of biological active substances. In determining the number of extractiles, it has been found that the maximum extraction endured the use of ethanol at concentration of 10, 20, and 30%. In the case of dry skinhorn fungus, the number of extractiles extracted at ethanol concentration of 10, 20, and 30 is 2 to 3 times greater than when using freshly picked mushroom. Polysaccharides are the main group of the basidomycetes, biological active substances. When determined the quantitative content of polysaccharides in dry materials, their content was found to be 12 to 28%. Comparing this value with polysaccharides of shiitake, maitake, reishi, cordyceps, and coriolus fungi, skinhorn fungus, in only inferior to shiitake. Tincture were prepared in the ratio of raw materials. The extract as 1 to 40 in term of dry matter, or 1 to 5 without taking into account the moisture content of the mushroom's food body. As extracts use a different concentration of ethanol from 10 to 18%. In the tincture, determine the content of polysaccharides, extractiles, identify their main group or base. In the identification of the main group of biological active substances, in the tincture using color reaction have been found polysaccharides, phthalate reagent, red precipitate, phenolic compounds, iron 3 chloride, brown color, steroid substances, sulfuric acid, concentrated pink color, amino acids with ninhydrate, blue, violet color, nitrogen contain compounds, alkaloids, Dragendorf reagent, orange color, and iridoids, star reagent, cyan color. Quantification of polysaccharides has been performed by a gravimetric method. The research results are shown on the slide. In tincture with raw materials, the maximum content of polysaccharides has been found in aqueous alcoholic solution of 20, 13, and 40%. Today on the internet on sale are dietary supplements with phallus and pudicus content. They are representing different release form: aqueous alcoholic tincture, capsules with mushroom dry powder, suppositorium, and dry mushroom. Producer of these dietary supplements have made the following recommendation for use as a treatment for malignant tumors, sarcoma, melanoma, leukosis, and other oncological disease. Benign tumor, prostate adenoma, ovarian cyst, polyps, hypertension, and for ulcer-healed are used externally. To sum up, we have completed the first step to determine chemical compounds of phallus and pudicus food body. We are intent to continue the science work, and I hope to introduce my results with you. My report is over. Thank you for your attention.
Thank you for report, Olga. Any questions for speaker?
I have a questions. Olga, please tell me, what are the prospects for using this extract, and what is the predicted pharmacological activity of it?
Okay. comparing this therapy with chemotherapy, which observes so many side effects, harmful side effects, treat with this tincture, mushroom tincture, no side effects were observed. And in my town, I'm from Rivne, even some oncological doctor also has recommended to try to find this mushroom, and with combined with basic therapy, with traditional therapy of oncological disease. So
Okay, understand. Thank you.
Thank you, Olga. I also have a question. Is it possible to grow this type of mushroom in Ukraine?
Yeah, of course, in Ukraine, but there's a little bit a problem, because this mushroom is so they hide from human eyes deep in the forest, but they grow in the summer, but it's so hard to find them in the forest. Yeah.
Thank you very much.
Thank you.
If no one has questions, next speaker is Sobolevska Evelina, PhD student from Lodz University of Technology, Faculty of Biotechnology and Food Sciences, Institute of Fermentation Technology and Microbiology, Lodz, Poland, with report Fungi attacking plants during cultivation and storage, food safety.
Can you see the presentation?
Yes, Evelina, we can see.
Okay. Good morning.
Oh,
My name is...
I'm sorry. We didn't
Your
see
mail.
presentation. We see your email.
Musisz zmienić na prezentację, bo pokazujesz skrzynkę pocztową.
Jak to?
Stop sharing the presentation and try again.
One minute, please.
It's okay.
And now?
Yes.
Yes, now we can see it.
Good. Okay. Good morning. My name is Ewelina Sobolewska. I am a PhD student at the Interdisciplinary Doctoral School of the Łódź University of Technology, Poland. I carry out my doctoral thesis under the supervision of PhD Anna Koziruk and PhD Anna Otlewska. The topic of my presentation will be fungi attacking plants during cultivation and storage in the aspect of food safety. Cereals, especially maize, rice, barley, and wheat, are the staple food source for the vast majority of the population, and at the same time are the target of pathogenic fungi. The infestation of cereals by fungi leads to a reduction in both their quality and quantity of cereals and huge economic losses. Cereal grains can become contaminated during cultivations, harvest, as well as storage and transport. Among the plant diseases caused by fungi, those affecting cereals pose a major problem from a food safety point of view. By 2050, the world's population is projected to increase by another 2 billion, and global food demand could increase by up to 50%. Yes. In addition, many fungal pathogens, including those of the genera Fusarium, Aspergillus, and Penicillium, are capable of producing mycotoxins that threaten the health of humans and animals. Considering food safety, the most important are deoxynivalenol, nivalenol, zearalenone, fumonizins, ochratoxin A, aflatoxins, T2 toxin, HT2 toxin, citrin, and patulin. Ochratoxin A and aflatoxins are produced by the storage fungi, Penicillium and Aspergillus, mainly after harvest. In contrast, deoxynivalenol, zearalenone, and fumonizins are produced by Fusarium fungi, mainly before harvest. Despite many studies over the years and the introduction of good production, and agricultural practices into the food chain, the presence of mycotoxins remains a global problem. The fungi that contaminate grains are divided into field fungi, storage fungi, and so-called intermediate fungi. The field fungi colonize grains in the field before harvest. Most of the field fungi produce hazardous mycotoxins both after and before harvest. This group includes, for example, species of the genera Fusarium and Alternaria. The next group is the storage fungi. The storage fungi may be present in small numbers prior to harvest. In addition, they can contaminate grains during harvest and increase their numbers as a result of storage, which may be affected by appropriate environmental conditions. The storage fungi primarily include, for example, species of the genera Aspergillus and Penicillium. The intermediate fungi continue their development during storage if water activity remains high. The intermediate fungal group includes, for example, species of the genera Fusarium, Cladosporium, and Trichoderma. Fusarium fungi infect many plant crops, including wheat, barley, triticule, rye, as well as maize, and many other plants important to humans. Plants can be infected at various stages of their development, causing pre- and post-emergency diseases. Infested crops are characterized by a reduced commercial and consumption value. Additionally, the obtained crops are contaminated with mycotoxins that are dangerous to humans and animals. Fusarium fungi produce zearalenone, deoxynivalenol, nivalenol, fumonizins, T2 toxin, and HT2 toxin. Cereal diseases are caused by various species of the genus Fusarium, mainly by Fusarium curmorum, Fusarium anaceum, Fusarium graminearum, and Microdochium nivale, formerly known as Fusarium nivale. They contribute, among others, for damage to ears, panicule, grain, foot, and crown of cereals. Various species of the genus Fusarium, mainly Fusarium graminearum, Fusarium curmorum, Fusarium moniliformis, Fusarium subglutinans, and Fusarium zaae, also damage maize. They contribute, among others, for damage to cobs, stalk bases, and roots of maize. Alternaria is the ubiquitous type of fungus that damages agricultural products, including cereal grains, such as wheat, barley, triticule, rye, and oats, both before and after harvest. Infection of cereals leads to a reduction in their quality. In addition to spoiling a wide variety of foods, several Alternaria species are capable of producing secondary metabolites considered to be phytotoxins, which play an important role in plant pathogenesis, and mycotoxins, which can be harmful to humans and animals. Cereal diseases caused by fungi of the genus Alternaria include black point and sooty molds. Black point is mainly caused by Alternaria and Cladosporium. Symptoms are only visible after harvest. Affected grain shows a darkening of the outer coat, particularly at the embryo end of the grain. Sooty molds is mainly caused by Alternaria and Cladosporium. The usual symptom of the sooty molds is a darkening of the ears before harvest. Fungi of the genera Aspergillus and Penicillium are mainly after harvest as a result of improper storage of the crop. They produce aflatoxins and ochratoxin A. Considerable damage to the grain, as well as its contamination with soil microorganisms, contribute to the high content of aflatoxins and ochratoxin A in the grain. The growth of molds and the mycotoxins produced both during storage and cultivation is undesirable. In the case of cereal production, this leads to contaminated grain. An attempt to comprehensively combat the sources of cereal diseases, from the moment of sowing seeds to vegetation of plants, harvesting of grains, and storing the crops, may reduce the development of fungal pathogens and thus ensure food safety. Thank you for your attention.
Thank you very much, Evelina. Any questions for Evelina? Adam, do you have a question?
No, thank you.
Okay, so next speaker is Koskar Julia, PhD student from Estonian University of Life Sciences, Institute of Veterinary Medicine and Animal Sciences, Chair of Food Hygiene and Veterinary Public Health, Tartu, Estonia, with the report The Prevalence and Concentration of Listeria monocytogenes in Ready-to-Eat Fish Products in Estonia.
Hello, everyone. My name is Julia Koskar, and my topic today is the prevalence and concentration of Listeria monocytogenes in ready-to-eat fish products in Estonia. Let me first of all shortly introduce you my university and a laboratory I work at. Estonian University of Life Sciences is the only university in Estonia whose priorities in academic and research activities provide the sustainable development of natural resources. It is one of top 50 universities in the world in the field of agriculture and forestry, and also top 1% most cited research facilities in the world. The Institute of Veterinary Medicine and Animal Sciences is composed of 10 departments that perform high-level modern teaching, research, and development activities in many fields. The chair of the basic and applied research activities of the chair of food hygiene and veterinary public health are focused on microbiological and chemical food hazards and reducing veterinary public health risks. Besides the topics related to zoonotic pathogens, research has been aimed at the antioxidative and antibacterial properties of food plants and their metabolites in relation to human health, which is actually the main topic of my PhD. I also work at Estonian Veterinary and Food Laboratory, which is a state agency operating in the area of government of the Ministry of Rural Affairs. It is accredited and offers high-quality laboratory services. In cooperation with the universities and other research institutions, both at home and abroad, we participate in research and development projects and act as a national reference laboratory. Now let's start with the topic, and first of all by introducing Listeria in general. Genus Listeria has 21 species. Out of the species identified so far, only Listeria monocytogenes can cause infections in both humans and animals. Listeria monocytogenes has been found widely in nature. Its growth conditions contribute to their ability to grow and survive under extreme environmental conditions, both at food processing facilities, and become a serious problem for food industry and thereby endangering the human health while being a causative agent of listeriosis.
Julia, I'm sorry, you didn't switch on your slides.
Can you see me switching it?
No,
No, okay, sorry.
you are not moving it. So like, yes, that's good.
Like that?
Yes, yes.
Thank you.
Thank you.
A little bit about listeriosis. It is a zoonotic disease that is mainly acquired through consumption of contaminated food and is characterized by a wide spectrum of infections, which are categorized into two forms, namely severe invasive listeriosis and non-invasive. Non-invasive form is the mild form of the disease, affecting mainly otherwise healthy people. Invasive listeriosis is a more severe form of the disease and affects certainly high-risk groups of the population. And this form of the disease is characterized by severe symptoms and high mortality rate. According to EFSA report, Listeria was in top 5 of zoonoses in the European Union in 2019, with a notification rate 2,621 per 100,000 population. In the European Union, the listeriosis cases have shown the increasing trend. In 2019, there were 2,621 confirmed human listeriosis cases with 21 outbreaks and 349 outbreak-related cases, which is 190 more than a year before. Among those 2,621 cases, there were 300 reported deaths with case fatality 17.6%, which is very, very high. The potential high-risk products for listeriosis are vacuum, packaged, sliced, cold, smoked meat and fish products, then other ready-to-eat fish and meat products with a long shelf life, raw fish and meat products, raw milk, also soft cheeses with higher moisture content, for example, feta, brie, camembert, other dairy products made unpasteurized milk, and also sliced or shredded, vacuum packed and modified atmosphere packed fruit and vegetables with a long shelf life. Now let me introduce you the Estonian study from years 2012 to 2016. The samples were collected from Estonian food business operators in the frame of self-control programs or by the state surveillance officers. Almost all Estonian analysis for Listeria monocytogenes in ready-to-eat food was included. Within 5 years in total of 30,016 ready-to-eat food samples were analyzed, and 3.6% were found to be positive for Listeria monocytogenes. Highest prevalence among food categories was found for ready-to-eat fish and fish products with 11.6%. Enumeration of Listeria monocytogenes was performed for 14,342 ready-to-eat food samples, and official food safety criterion, which is 100 colony forming units per gram, was exceeded for 46 samples, which was 0.3%. Comparing to 2% prevalence of Listeria monocytogenes determined in ready-to-eat foods in the previous study in from 2008 to 2010, the overall prevalence in ready-to-eat foods determined in the present study was higher and was mostly contributed with high proportion of Listeria monocytogenes positive samples among ready-to-eat salted and smoked fish products. Now, a little bit about Listeria monocytogenes specifically in ready-to-eat fish products. The study showed that the highest prevalence among products were for salted fish and fish products, with a percentage of 28.6. These products were mostly lightly salted, which means with a final sodium chloride content of 1.3 to 1.5. The percentage of cold smoked fish products was also high, 26.5. This study has been published in the Journal of Food Protection in 2019. In conclusion, I would say that Listeria monocytogenes is an issue in Estonia among ready-to-eat foods, and more effective control strategies are needed to reduce the contamination in problematic food business operators in Estonia.
Thank you very much, Julia. Any questions?
Yes, I have a question for you.
Yes.
So tell me please, what are the parameters of qualitative and quantitative control of fish products in your country, in Estonia, except the concentration of Listeria monocytogenes?
Do you mean?
I mean, how do in Estonia control fish products?
Mm-hmm. Okay, so one criteria is that if your product is with a short shelf life, which is less than 5 days, then actually Listeria is not an issue. It's actually when you have vacuum packed or somehow packed products with a long shelf life. Then you need to check for Listeria. Then you can have it tested. It's either the zero tolerance, which means no Listeria can be found in a product, or if you can demonstrate to the authority that your product doesn't exceed the concentration, the 100 colony forming units at the end of shelf life, then it's enough.
Okay, thank you. Thank you for your report. Thank you very much.
Thank you, Julia. The next speaker is Piekarska-Radzik Lydia, PhD student from Lodz University of Technology, Faculty of Biotechnology and Food Science, Institute of Fermentation Technology and Microbiology, Lodz, Poland, with the topic Bacteria of genus Lactobacilliaceae and plant polyphenols as an alternative to antibiotic therapy in Staphylococcus infectious, a review.
Hello everyone, my name is Lydia Piekarska-Radzik. I am a PhD student at Lodz University of Technology at the Faculty of Biotechnology and Food Sciences at the Institute of Fermentation Technology and Microbiology. And during my presentation, I would like to show you the possibility of limiting the growth of bacteria of genus Staphylococcus by means other than antibiotics. And this is also the topic of my PhD thesis. Staphylococcus bacteria were first described by Alexander Oakston. Currently, this group includes as many as 47 gram-positive immobile and non-sporulating species. Staphylococcus bacteria can be found in virtually any environment, such as water, and animal products. So not wonder that they colonize the entire surface of the human body, starting with the scalp and ending with the feet. However, it is worth mentioning that there are pathogenic species such as Staphylococcus aureus. Staphylococcus aureus infection may not only cause local infection, mainly skin and subcutaneous tissue infections, which are characterized by the presence of purulent discharge. But it also causes invasive infection with a high mortality, such as sepsis and endocarditis. At this point, one might ask why I am bringing up the issue of antibiotic resistance today. As an example, I would like to show you the results of some research. In 2013, research on secondary nosocomial infection was carried out in one of the hospitals in Szczecin. In a group of 100 randomly selected patients undergoing head surgery, 14 developed a bacterial infection, and it is noteworthy that as many as 9 out of 14 cultures from the patient wounds revealed the presence of Staphylococcus aureus bacteria, including methicillin-resistant Staphylococcus aureus. It is the increased resistance of Staphylococcus aureus strains to antibiotics that is one of the very serious epidemiological problems of the modern world. Before the use of penicillin, the mortality rate of human with Staphylococcus aureus infection was up to 70%. The use of penicillin reduced the mortality from infection to less than 30%. Unfortunately, within 10 years, Staphylococcus aureus further increased mortality by almost 20%. The effect of using next antibiotic like methicillin was analogous to the penicillin. The initial decline in mortality resulted in the development of the antibiotic resistance. Currently, in addition to penicillin and methicillin resistance, strains that are resistant to glycopeptides or other antibiotics routinely used to treat MRSA infection are also being isolated. Therefore, it is important to find substances of natural origin that have bactericidal potential and could help fight bactericidal infection that are difficult to suppress. For this purpose, the lactic acid bacteria described by Orla Jensen can be used. It includes microaerophilic microorganisms. They are usually immobile, gram-positive, non-spore-forming bacilli, and their main characteristic is ability to ferment sugar, mainly into lactic acid. But in addition to lactic acid, those bacteria are able to synthesize other carboxylic acids, such as acetic acid or propionic acid. Apart from that, byproducts of the lactic acid fermentation process can be, for example, carbon dioxide or diacetyl. Moreover, lactic acid bacteria are able to synthesize other specific compounds such as deuterin, deuterocycline, or bacteriocins. And it is thanks to this wide range of synthesized metabolites that lactic acid bacteria have the antagonistic properties that form the basis of my today's presentation. Let's start. Lactic acid is primarily metabolite of lactic acid bacteria. It is a key compound responsible for antagonistic activity of those bacteria. According to the literature data, lactic acid of concentration 0.5% is able to limit the growth of pathogens such as Staphylococcus aureus. The antimicrobial action of lactic acid is mainly based on condensation on DNA molecules and abnormal replication processes. Impaired synthesis of extracellular protein can lead to cell death. Bacteriocins are metabolites, mainly protein ones, produced by lactic acid bacteria that have high bactericidal potential. The advantage of bacteriocins synthesized by lactic acid bacteria over antibiotics is their stability in high temperature and resistant to stress related to pH change. It is worth emphasizing that bacteriocins synthesized by lactobacillus strains are safe for human health. Therefore, they are usually used in food industry. Another metabolite produced by lactic acid bacteria is hydrogen peroxide. Hydrogen peroxide is a bactericidal agent used in many industries such as medicine, food industry, and environment protection even. This compound is used to disinfect many surfaces, medicine devices, and water treatment. Hydrogen peroxide, which is non-toxic to the environment, has the ability to antagonize the action of bacteria endospores. And the cytotoxicity of hydrogen peroxide is related to the ability to convert to more reactive forms very strong oxidant, such as the hydroxyl ion. The bactericidal activity of oxidant is often based on the ability to cause damage proteins, lipids, and nucleic acids. Moreover, hydrogen peroxide can increase the oxidative stress in bacterial cells. Deuterin is characterized by a broad spectrum of antagonistic activity against gram-negative or gram-positive bacteria. It is water-soluble compound. It has bactericidal activity in wide range of pH. During the research on deuterin, one more substance synthesized by lactic acid bacteria was identified, deutericycline. Deutericycline also has a very broad spectrum of influence on bacteria, considered pathogenic, mainly among gram-positive bacteria, and Staphylococcus aureus is one of the most typical representative of this group of microorganisms. Plant extracts have been used by humans for countries, not only in the food industry, but above all in medicine. The possibility of using plants in traditional or modern medicine is related primarily to the high content of polyphenols in their composition. Polyphenols are plant metabolites that have a protective function, for example, antifungal. They are stored in various parts of plants, such as fruit, flowers, leaf, or stems. But in the human body, polyphenols induce a number of changes. They show antioxidant activity. They have anti-allergenic or blood pressure lowering properties. They can be anti-diabetic and anti-inflammatory. Above all, however, they have antimicrobial potential, and it is the feature that I will investigate. The bactericidal activity of polyphenol is primarily due to their ability to cause ruptures in bacterial cell membranes, then may cause leakage of cellular compounds such as ATP, DNA, proteins. Accordingly, the cellular metabolism changes, and consequently, it leads to cell death. Summarizing, I believe that the activity of metabolites synthesized by lactic acid bacteria and polyphenols extracted from plants can effectively reduce the growth of bacteria from the genus of Staphylococcus. It is worth noting that the antimicrobial properties of lactic acid metabolites and polyphenols have a very similar effect on pathogen cells. Since both factors primarily induce ruptures in pathogenic cell membranes, an alternative way of limiting their growth can thus be obtained. This is references from my presentation. And if you are interested in my research, I would also like to encourage you to take a look at the review article I prepared. In the article, I focused also on the interaction between polyphenols and fermenting bacteria in food. For example, the prebiotic property of polyphenols. Now I would like to thank you for your attention. And if you have any questions, please contact me using this email address. Thank you very much.
Thank you, Lydia, very much. Questions for speaker? No questions. So next speaker is Копытник Екатерина Владимировна, лаборант отраслевой лаборатории ДНК и клеточной технологии в растениеводстве и животноводстве, Полесский государственный университет, город Пинск, Беларусь. С докладом «Павловния войлочная. Введение в культуру инвитро, культивирование и адаптация».
Здравствуйте. Также я являюсь студентом Полесского государственного университета биотехнологического факультета, на базе которой расположена данная лаборатория. Тема исследования – Павловния войлочная, введение в культуру инвитро, культивирование и адаптация. Павловния войлочная – дерево, отличающееся высоким хозяйственно-биологическим потенциалом. Представляет интерес как декоративная и ценная древесная порода. В связи с этим возникает потребность в ее производстве его плантационных количеств. Это можно достичь только с помощью микроклонального тиражирования, то есть размножения в условиях инвитро. Цель работы – проращивание и размножение в условиях инвитро семян павловнии войлочной, а также ее адаптация к условиям экс-витро. Для ввода стерильных семян в культуру инвитро были использованы растворы фунгицидов Ридомил Голд и Байдан по 200 мг на 100 мл раствора с добавлением 2 мг аскорбиновой кислоты и 300 микролитров Твин-20 из расчета на 100 мл раствора. Время экспозиции 18 минут. И стерилизующий агент 7,5% раствор гипохлорида кальция. Время экспозиции 25 минут. Проростки, полученные из стерильных семян, высаживали на питательную агаризованную среду Мурасигис куга с 6-бензилоаминопурином и среду Андерсона. В дальнейшем растения были разделены на экспланты и пересажены на ту же самую среду с добавлением гормона. Банки с семенами размещали на стеллажах световой установки культурального помещения биотехнологической лаборатории при температуре плюс 25 градусов, в фотопериоде день-ночь 16/8, освещением 4000 люкс и относительной влажностью воздуха 70%. Учет количества проросших семян проводили через каждые 10 дней в течение 4 месяцев культивирования на стеллажах световой. Стерильные семена начали прорастать на 6-й день от начала эксперимента, и их схожесть на 10-е сутки составила 76%, в отличие от нестерильных семян, которые начали прорастать на 2-е сутки со схожестью 33%. Показатели роста и активности эксплантов к пролиферации на среде Андерсона оказались значительно ниже, чем на среде Мурасигис куга. В среде Андерсона изначально есть предшественник цитокина 6-бензилоаминопурина, а в среду Мурасигис куга добавляли 6-бензилоаминопурин в концентрации 1 мг на литр. Рост и развитие на среде Мурасиги-Скуга идет лучше. Дальнейшее черенкование основного и дополнительного побегов инвитро обеспечило коэффициент размножения в первом цикле на среде Мурасиги-Скуга 1 к 8, а на среде Андерсона до 1 к 4. Далее, в период адаптации стерильные растения были высажены в грунт длиной в среднем 15,9 мм, а нестерильные длина их составляла 4,09 мм. Средняя высота высаженных в грунт и культивируемых на стеллажах адаптационного помещения стерильных растений на каждый день замера превышала таковую у нестерильных. Максимальная высота стерильных адаптантов достигала 35 мм, и максимальный прирост составил 18 мм, в то время как у нестерильных адаптантов максимальная высота составила 9 мм, а прирост не превысил 5 мм. На основании проведенных исследований можно сделать выводы. Схожесть стерильных семян побловни и волочной составила 76%. Среда Мурасиги-Скуга с добавлением гормона БАП в концентрации 1 мг на литр является оптимальной для культивирования эксплантов, полученных из семян. Наиболее высокий коэффициент размножения наблюдается на среде Мурасиги-Скуга, и максимальная высота и скорость прироста стерильных адаптантов значительно выше, чем нестерильных, что говорит о положительном влиянии этапа стерилизации. У меня все.
Спасибо большое за доклад, Екатерина Владимировна. Есть ли вопросы к докладчику? Екатерина Владимировна, у меня есть вопрос. Сейчас павловния также очень популярна в Украине, но есть ли у вас информация, насколько сильно эти деревья истощают почву? Потому что растут они очень быстро, и крона достаточно массивная.
Дело в том, что у них стержневая корневая система, и она проникает очень глубоко в недра земли, поэтому, что касается именно плодородного слоя почвы, это будет не так сказываться на наземных растениях, потому что все, что берется, оно берется из глубины, не из поверхностного слоя земли.
Спасибо большое.
Еще можно вопрос?
Да, конечно.
Будьте добры, Екатерина, очень интересный доклад у вас. Скажите, пожалуйста, данное растение используется больше я прочитал, что для получения древесины, оно очень культивируется как красивое растение, очень красивые цветки. Вы не встречали информации по использованию его для получения каких-то экстрактов, для применения как лекарственные препараты? Не было у вас, не встречали вы такую информацию?
Она применяется для косметических средств, там есть полезные кислоты, которые применяются в косметологии. Насчет экстрактов фармацевтической промышленности я затрудняюсь ответить на этот вопрос.
Ну, тогда я думаю, что мы сможем с вами совместно поработать в этом плане, поработать либо вы с кафедрой биотехнологии нашего университета, либо с нашей кафедрой технологии фармпрепаратов, поработать над созданием лекарственных препаратов, возможно, с этого растения, с этого дерева. Спасибо большое за доклад, очень хороший.
Спасибо и вам.
The next speaker is Kuus Kaisa, PhD student of Estonian University of Life Sciences, Tartu, Estonia, with presentation Salmonella in meat chain and Salmonellosis in Estonia. Kaisa? Are you here?
Yeah, so do you see my screen?
Yes, we can see your screen, but not your presentation.
Right now?
Yeah, that's good.
All right. So, hello everyone, and my name is Kaisa Kuus, and I'm PhD student at the Estonian University of Life Sciences, and also I work at the Estonian Health Board. And I'm going to speak a little bit about Salmonella in meat chain and about salmonellosis in Estonia. The aim of this study was to determine the most prevailing Salmonella serotypes in Estonian meat chain and regarding to human salmonellosis cases. Study will continue in future with molecular characterization by using bioinformatic analysis. On this slide is presented taxonomy of Salmonella bacteria. The bacteria belongs to Salmonella family. There are two species of Salmonella. Salmonella enterica and Salmonella bongori. In Salmonella enterica species, there are 6 subspecies, as you can see here. Also, according to Kaufman-White scheme, there are over 2,500 serotypes of Salmonella. Also, there are many Salmonella serotypes, but some of them are adapted to infect human. For example, Salmonella typhi and Salmonella paratyphi. On the other hand, some serotypes are adapted to infect particular animal species, but also there are serotypes that are adapted to infect both animals and humans. According to European Food Safety Authority, the most common zoonosis in European Union in 2019 is Campylobacter, followed by Salmonellosis. I think you already saw this slide before, but here it is again. And on this chart, you can see that there has been a little decrease in Salmonella cases from 2010 to 2019. The number of cases has been pretty stable in last years. Maybe it's because of that the nation's regulations have been so good that they have it under control, basically. As were said before, Salmonella is responsible for many foodborne illness cases on humans. In 2019, there was 87,923 cases of Salmonella, and hospitalized were 16,628 patients, and reported deaths were 140. It means that the case fatality is basically 0.2. And now, according to Estonian Health Board data, in 2018, there were 323 Salmonella cases in Estonia. Next year, on 2019, there were 154 cases per year, and last year there were only 92 cases. Maybe it's because people were traveling less last year or going out eating less than they used to because of COVID situation in world. On this slide is presented Salmonellosis and Campylobacteriosis cases in Estonia from 2010 to 2019. As you can see, this chart, Salmonella cases have been decreased, and Campylobacter cases, on the other hand, have been increased. On this chart, we can see the prevalence of different Salmonella serotypes in humans. The most common serotype isolated from human patients were Salmonella enteritidis. It's here with blue. And also there were many cases with Salmonella typhimurium, and it's with orange. And some serotypes like Salmonella derby that are pretty common in animals are rarely isolated from humans. And according to Estonian Health Board data on 2019, 91% from all the human cases were caused by Salmonella enteritidis, as you can see here, and 31% was caused by Salmonella typhimurium and its monophasic variant. Next year, on 2012, the picture is pretty same because 46.7% cases were caused by Salmonella enteritidis, and 25% cases were caused by Salmonella typhimurium and its monophasic variant. And the main Salmonella sources on this slide, Salmonella enteritidis and Salmonella typhimurium are the most important Salmonella serotypes in medicine that's involving both human and animal infections. The main sources are eggs and poultry meat, raw milk and contaminated dairy products, pork, beef and game meat, fish and shellfish, fresh salads, and of course contact with ill people and traveling. And now I'm going to speak a little bit about Salmonella in slaughterhouses and in meat cutting level in Estonia. Here are presented different animal species and number of positive samples from 2015 to 2019. And as you can see, the most positive samples were isolated from swine carcasses, and from the others, it was less than from swines. And in meat cutting level, we can see similar results that the most of the positive samples are from pigs. On this slide, you can see which serotypes are most common in slaughterhouses and in meat cutting level. But also, you can see how commonly are different serotypes isolated from different meats. The most common serotype that has been isolated is Salmonella derby, and as you can see, mostly it is found from swine carcasses or from its meat. And on this chart, you can see the occurrence of Salmonella typhimurium and its monophasic variant in meat production chain from 2013 to 2018. Over the years, there have been increase in number of samples that are positive for typhimurium. And as you can see in 2018, there has been an incredible increase in Salmonella typhimurium's monophasic variant. And on this chart, you can see the serotype distribution of Salmonella enterica, subspecies enterica, isolates originated from meat production chain. The most common serotypes from meat production chains are Salmonella terbi, followed by Salmonella typhimurium and its monophasic variant. But as you remember, I previously showed you about human cases, and there was said that from human there are mostly isolated Salmonella enteritidis, but here it is not so common in animals as it were on humans. And Salmonella terbi, which is the most commonly isolated serotype from meat production chain, from human cases, it was rarely isolated. And thank you for your attention.
Thank you very much, Kaisa. Questions for speaker? No questions. So the next speaker is Szczeszka Silvia, PhD student from Lodz University of Technology, Lodz, Poland, with the report Algae as an ingredient of innovative functional food with probiotic features. Silvia, we didn't hear you. No, please switch on your microphone.
Yes, sorry.
Yeah, it's okay.
Great. Yes, now can you hear me and see my presentation?
Yeah, everything is perfect.
Yes, thank you. I'm really sorry. Okay, so good morning or good afternoon. First of all, let me thank you all for coming here today. I would like to introduce myself. My name is Sylwia Ścieszka, and currently I'm a PhD student in the Institute of Fermentation Technology and Microbiology at the Lodz University of Technology in Poland. My interests are connected with lactic acid bacteria, diet, functional food, and algae. That is why I would like to talk to you today about algae and lactic acid bacteria, so everything that is needed to create healthy fermented food products. First of all, I will explain why algae are worth consuming. I will continue by giving examples of food products with algae. To finish, I will offer some solutions how to create an innovative fermented products with algae. If you have any questions, feel free to interrupt me at any time. Okay, so let's begin with the algae. Did you know that algae are some of the most common organisms inhabiting the earth? They are able to grow even in extreme conditions. They are present in terrestrial and aqueous environments, and they grow both in fresh and salt waters. The internet database AlgaBase describes over 160,000 species. During my PhD, I used powdered Chlorella vulgaris from Bailey's food. Let's move on and discuss what properties have algae. Algae produce a broad spectrum of bioactive secondary metabolites and valuable bioactive substances, including lipids, polyphenols, polysaccharides, sterols, pigments like chlorophyll, carotenoids, or phycobilins. Moreover, algae are a rich source of elements, vitamins, and dietary fiber. Due to their properties, biological activity, rich chemical composition, and content of bioactive substances, algae may be used for construction of functional food products. Algae have been used as food since ancient times. In the Far East and Asia, the tradition of eating algae is a long-standing one, whereas in Western countries, the interest in alga-based products is quite recent. Algae are very popular among vegetarians who use them as starters, traditions, and main courses. Let's move on and discuss what food products contain algae. In food for human consumption, algae have been mostly introduced to meat products, such as steaks, frankfurters, and sausages, as well as to fish products, and oils. Another group of products enriched with algae are cereal-based products, such as pasta, bread, and flour. Attempts were also made to create an innovative functional product with algae, such as hamburger with algae. Nevertheless, some aspects, including quality and organoleptic acceptance, still require more detailed analysis. Nowadays, customers demand novel products, and interest in algae has increased significantly. Several companies have gotten involved in developing foods containing microalgae. Some alga species, such as chlorella, which has a great potential for human diet and falls into the category of generally recognized as safe, are already commercially used in food. Examples of food products with chlorella include organic alga drink from Halga brand, fresh slow juice, algae crackers from Halga, baked bean crackers from Jinbi, crispy matcha biscuits produced in Japan, super gig bar produced in Germany, sesame slices with chlorella, and chlorella fudge produced by Miami in Poland. But what about fermented products containing algae? Popular fermented products enriched with chlorella are mainly dairy products, such as probiotic fermented milk and various types of cheese like Appenzeller cheese. Nevertheless, only a few studies have focused so far on the practical use of algae and fermented food. The most important microorganisms used in food fermentation are lactic acid bacteria. From the industrial point of view, they are a very important microorganism. They provide an effective method of conservation of fermented food, affects its taste, texture, and often also the nutritional value. Composition of lab-rich products is associated with numerous health benefits. However, have you ever felt disappointed during shopping in the supermarket when you wanted to buy healthy fermented products? I've been working to design innovative functional food products with algae and lactic acid bacteria as to make sure to fill the gap in the food industry. I investigated the impact of the Chlorella vulgaris on the growth and acidifying activity of lactic acid bacteria. Then I examined the probiotic features, such as survival in adverse environmental conditions, in the presence of sodium chloride, bile salts, in phenol, and at low level of pH. Furthermore, I checked the impact of Chlorella vulgaris on the antibiotic resistance and antagonistic activity. The results revealed that the combination of Chlorella vulgaris and Levylactobacillus brevis shows great potential for creating innovative functional products that can provide a significant amount of lactic acid bacteria to the customer, in addition to the nutritional properties of algae. Therefore, I've been creating new innovative fermented products with algae. I'd like to elaborate on the first innovative product which I made. I added powdered Chlorella vulgaris from Bali's food and lactic acid bacteria Levylactobacillus brevis WOTSC 944. It's from Pure Culture of Collection Microorganisms WOTSC 105 at the Lodz University of Technology in Poland, and added it into the soya drink. And that's how I create an innovative probiotic fermented soya drink with algae. The survival of lactic acid bacteria in soya drink with and without algae was tested by the play method during the fermentation process, maturation during 30 days of storage, and gastrointestinal tract. Results obtained in this study revealed that the soya drink and Chlorella vulgaris were a sufficient source of medium for Levylactobacillus brevis. Moreover, due to the number of bacteria and survival during the storage and also after gastrointestinal tract, this soya drink with Chlorella vulgaris meets the criteria for fermented probiotic products. Furthermore, the results are patented as a pattern application for the innovation, a method for producing a probiotic fermented soya drink with algae. To wrap up, let's remind ourselves of why this should matter to everyone here. Combination of fermented products offering a high content of lactic acid bacteria with algae possessing a biological active metabolites of natural origin allows to compose products with a high content of nutrients and to create a brand new segment of fermented food. Thank you for attention. If you have any questions, I'll be happy to answer them.
Thank you very much, Sylvia. Questions for speaker? No questions? Thank you very much. So the next speaker is Києнко Людмила Сергіївна, аспірант кафедри заводської технології ліків з Національного фармацевтичного університету Харків, Україна, з доповіддю на тему допитання про герпевірусну інфекцію як актуальну проблему сьогодення.
Одну хвилинку.
Так, Людмила, чекаємо.
Доброго дня. Тема моєї доповіді – допитання про герпесвірусну інфекцію як актуальну проблему сьогодення. Термін герпес має давньогрецьке походження. Герпо у перекладі означає той, який стелиться, повзе. 2400 років тому так назвав хворобу Гіппократ. По-перше, симптоми герпесу описані римським лікарем Геродотом у 5 столітті до нашої ери. В 1736 році лікар короля Людовика XV Астрюк у книзі «De morbis venereis» описав генітальний герпес. Вірусну природу герпесу експериментально довели у 1912 році німецькі вчені Грютер та Левенштейн. Вони змоделювали герпетичний кератит у кролів при нанесенні на рогівку ока вмісту пухирців, отриманого від хворого простим герпесом. Першу культуру інвітро вірусу простого герпесу було отримано у 1920 році. З того часу відкрито 8 антигенних серотипів вірусу герпесу та доведено їхню роль в етіології понад 40 захворювань людини. Класифікацію герпес-вірусів запропонував Ендрюс у 1954 році. Герпес – це одна з найрозповсюдженіших вірусних інфекцій людини. За даними Всесвітньої організації охорони здоров'я, близько 95% населення земної кулі інфіковані одним або декількома типами вірусу герпесу, але тільки 10% мають клінічні симптоми. Щорічно кількість хворих на герпес зростає, а смертність займає друге місце після грипу. Рецидиви герпесу спостерігаються у 20% пацієнтів. Частота прояву герпесу на губах може повторюватись від 2 до 12 разів на рік. Встановлення кількості нових випадків захворювання на герпес в Україні залишається доволі складним завданням. Це пов'язано з відсутністю обов'язкової реєстрації пацієнтів. Згідно зі статистикою, кількість хворих на герпес-вірусні інфекції в Україні становить 120-200 осіб на 10 тисяч населення. Медико-соціальна загроза даного захворювання обумовлена декількома факторами. Герпес-вірусні інфекції досить часто мають латентну клінічну картину. Завдяки імуносупресивній дії герпес-вірусів відбувається збій імунної системи організму, що може привести до різноманітних ускладнень та хронічного перебігу захворювання. Герпес-вірусні інфекції характеризуються довічною персистенцією вірусу в клітинах хазяїна. Проблема герпес-вірусних захворювань пов'язана також з частими рецидивами, що в цілому призводить до зниження якості життя пацієнтів та постійного дискомфорту, до психологічних та емоційних реакцій. Віруси простого герпесу належать до родини герпес-віріде. На сьогодні відкрито близько 100 представників сімейства вірусів герпесу, з яких 8 типів є патогенними для людини. Залежно від типу клітин залучених в інфекційний процес, характеру реплікації вірусів, структури їх генома. Герпес-віруси ділять на 3 підродини – альфа, бета і гамма. Половина випадків хвороби Альцгеймера пов'язана з вірусом простого герпесу першого типу. Також люди, які протягом життя заразилися вірусом герпесу, 3 рази більше схильні до розвитку стенокардії. Вірус простого герпесу першого типу також приводить до ураження центральної нервової системи, очей, печінки, слизових оболонок, шкіри. Він може індукувати розвиток пухлин. Вірус простого герпесу впливає на розвиток дисплазії і карциноми шийки матки. Для злоякісного переродження клітин організму не обов'язкова постійна наявність вірусу в клітинах. Він може діяти за принципом hit and run. Вірус простого герпесу другого типу впливає на репродуктивну систему. Останнім часом все рідше зустрічаються інфекції, викликані лише одним типом вірусу. Найчастіше спостерігають мікс інфекції, що становлять близько 64%. Найбільш поширені віруси першого та другого типів, антитіла до яких виявляють у 90% населення. Серед етіологічних чинників інфекційного синдрому найбільш часто спостерігають бактеріальні та вірусні коінфекції. При інфекційно-запальному процесі, в переважній більшості випадків, має місце ураження органів верхніх і нижніх дихальних шляхів та шкіри. Алергічні реакції виявляють у 35% хворих на мікс герпес-вірусні інфекції. Синдром дисбіотичних порушень в 30% випадків. Значення мікробної флори для організму людини відоме давно. Доведена її участь у формуванні імунного статусу, загальної неспецифічної резистентності в процесах травлення тощо. Встановлено, що до герпетичної інфекції може приєднуватись вторинна бактеріальна або грибкова інфекція. Активація бактеріальної флори при вірусних інфекціях досягає 50%. Найбільш представлені комбінації стрептококової та лактобацилярної мікрофлори, що становить 37%. На фармацевтичному ринку України лікарські засоби для лікування герпес-вірусних захворювань мають бути представлені широким асортиментом та різноманітними лікарськими формами, що надає можливості лікарю і пацієнту обрати препарат з урахуванням місця ураження, ефективності та безпечності препарату і фінансових спроможностей хворого. З огляду на це важливо було дослідити сегмент препаратів для лікування герпес-вірусних захворювань на вітчизняному фармацевтичному ринку та визначити, які лікарські форми наявні у більшості, а які представлені обмежено. За даними Державного реєстру лікарських засобів України, на вітчизняному ринку представлено 80 найменувань лікарських препаратів для лікування вищезазначеного захворювання. Аналіз асортименту вказаних лікарських засобів за формами випуску наведено на рисунку. Найбільш розповсюдженою лікарською формою є таблетки, що становить 70%. Також на фармацевтичному ринку України присутні сиропи, порошки та капсули. Краплі 2%, ліофілізат для розчину для інфузії 1%. Частка м'яких лікарських форм у досліджуваній групі становить лише 8%, а саме у формі крему 6% та мазі 2%. Асортимент лікарських препаратів вітчизняного виробництва у таблетованих формах представлений наступними торговими найменуваннями: ацеклавір Астраформ, ацеклавір Фармак, ацеклавір Дарниця, ацеклавір, герпевір, валавір, вальтравір та інші. Сиропи наявні такими торговими найменуваннями: неоприназин, новірин, флавозід. Наступну позицію займають капсули, що становить 4%. Це нуклекс. 3% складають краплі, протифлазід. Вітчизняні лікарські засоби у формі порошків представлені препаратами герпевір. Асортимент групи м'яких лікарських засобів представлений у формі мазі герпевір та крему агерб. Асортимент лікарських засобів іноземного виробництва у формі таблеток представлений наступними торговими найменуваннями. Це Зовірокс, Оцик, Ациклавір-Стода, Віролекс, Гівірон, Медовір та Волцик. Наступну позицію після таблеток займають креми та сиропи по 5%. У формі крему представлені такі торгові найменування: Оцик, Зовірокс, Ациклостод. Сиропи наявні такими найменуваннями, як гропринозин Ріхтер, неопринозин, Нормомед. Лікарські засоби у формі порошків становлять 2%, Віролекс та Медовір. Також на фармацевтичному ринку України наявні ліофілізат для виготовлення розчинів для інфузій, Зовірокс та сиропи, гропринозин, Ріхтер, неопринозин та Нормомед. Найбільш зручними і прийнятними у лікуванні герпес-вірусних хвороб є м'які лікарські форми. Вони мають низку переваг у порівнянні з іншими лікарськими формами. Це здатність забезпечувати тривалу концентрацію діючих речовин безпосередньо у місці нанесення препарату, простоту у застосуванні, можливість поєднання декількох діючих речовин різного фармакологічного спектру дії, забезпечення комплексного підходу до лікування тощо. Трансдермальний шлях уведення лікарських засобів дозволяє знизити ефект пресистемного метаболізму в печінці, не залежить від pH-вмісту шлунку, часу приймання їжі, можливостей метаболічної інактивації. Надходження лікарських препаратів через шкіру виключає можливість передозування та частоту прояву побічних дій, що часто виникають при пероральному шляху введення. Асортимент м'яких лікарських засобів, представлених на вітчизняному ринку, наведений в таблиці. Аналіз асортименту препаратів для лікування герпетичних захворювань показав, що за кількістю лікарських засобів перше місце належить Україні, що становить 51%. Пропозиції на фармацевтичний ринок надходять також з Німеччини, Польщі. Фармацевтичні виробники Угорщини постачають 5% лікарських засобів, Словенії, Кіпру, Росії, Італії по 4%, Канади, Великої Британії та Румунії по 1%. В результаті аналізу асортименту лікарських засобів за комбінацією діючих речовин встановлено, що домінують однокомпонентні препарати 94%. Комбіновані засоби займають лише 6%. За результатами ситуаційного аналізу розроблено асортиментний контур сегмента українського фармацевтичного ринку лікарських препаратів, які застосовуються для лікування герпес-вірусних захворювань. Таким чином, проведений аналіз асортименту протигерпетичних засобів свідчить про перевагу однокомпонентних препаратів і недостатню кількість лікарських засобів у м'яких лікарських формах. Отже, розробка комбінованої м'якої лікарської форми для лікування герпес-вірусних захворювань є перспективним напрямком у розвитку вітчизняної фармацевтичної науки та промисловості. Тому найбільш доцільним для досягнення максимального терапевтичного ефекту є створення лікарського засобу на основі субстанцій, що виявляють різну направленість дії, до перспективних активних фармацевтичних інгредієнтів у лікуванні герпес-вірусних захворювань можна віднести ациклавір та мірамістин. Перевагами ациклавіру є висока вибірковість до вірусів герпесу, низька токсичність, відсутність тератогенного і мутагенного ефектів, відсутність впливу на мікрофлору кишечнику, безпечність при тривалому застосуванні. Застосування ациклавіру при герпес-вірусних захворюваннях попереджає появу нових елементів висипу, прискорює утворення кірочок, послаблює больові відчуття. Мірамістин відноситься до групи катіонних поверхнево-активних речовин. Він володіє антимікробною дією по відношенню до грам-позитивних, наприклад, до стафілококів, стрептококів, та грам-негативних – гонококи, сальмонели, хламідії, мікобактерії. бактерій та грибів Candida albicans, Aspergillus, Penicillium, Trichophyton rubrum тощо, а також вірусів найпростіших. Мірамістин підвищує захисні реакції за рахунок активації фагоцитів, прискорює процеси регенерації та епітелізації, чинить протизапальну дію за рахунок активації фібринолізу в осередку запалення. Він не виявляє місцево-подразнювальної, мутагенної, канцерогенної, ембріотоксичної дій, не викликає алергії та має низьку токсичність, не чинить імуноадювантну дію. Таким чином, комбіноване використання декількох субстанцій в одній лікарській формі дозволить комплексно впливати на різні ланки патогенезу хвороби і тим самим прискорювати процес одужання хворого. На висновках дозвольте не зупинятись. Дякую за увагу.
Людмила Сергіївна, дуже дякуємо за вашу презентацію.
Дякую.
The next speaker is Яшневська Агнешка, перепрошую, with the topic of simplified methods for bakery starter culture production. Люда, пожалуйста, выключите демонстрацию экрана. Люда, сверху кнопочка. Дякую. Agnieszka, do you hear us?
Yes, here I am.
That's good.
Just a second. I have to find my presentation.
Oh, it's okay.
Oh, there it is. Okay, please let me know if you can see.
Yeah, we can see it. Thank you.
Okay, so let me introduce myself. My name is Agnieszka Jaśniewska, and today I would like to present you the topic of simplified method for bakery starter culture production. I am from Łódź University of Technology. I work in the Institute of Fermentation Technology and Microbiology in Łódź in Poland. First, I would like to start with emphasis on effect of sourdough on nutritional bread quality. Lactic acid bacteria that is present in the sourdough possess a proteolytic activity which causes degradation of wheat proteins such as gluten, which is responsible for celiac disease. Including sourdough in a bread recipe gives a chance to produce cereal products that could be well tolerated by celiac patients or people allergic to wheat proteins or gluten. Biofortification increases the content of nutrients such as vitamins or bioactive compounds. Reduction in phytate content, which is present in cereals and also acts as anti-nutrient, which prevents absorption of dietary nutrients in our digestive system. Lactic acid bacteria enable to dephosphorylate the phytic acid, which lowers or eliminates its binding affinity, and as a result supports bioavailability of minerals. Some exopolysaccharides undergo biosynthesis during sourdough fermentation, and they can act as prebiotics, positively affecting our digestive system. During sourdough fermentation, numerous aroma compounds are released, and they contribute to the rich sensory profile of sourdough bread. It is not always possible to use fresh sourdough in your recipe. That's why some bakeries use dry sourdough. Dry sourdough is obtained from mature sourdough and then stabilized by freeze drying, spray granulation, fluidized bed drying, drum drying, or the most popular method, which is spray drying. Depending on the technique applied for drying, we obtain different sensory profile. Different key flavor compounds are produced during drying. As well, we obtain different physical and chemical characteristics of our sourdough. Physical characteristic, they would affect powder behavior during storage and handling, while chemical properties, especially acidity, affect technological and sensory profile of our product. While it is necessary for a starter culture to contain active microorganisms, shelf-stable product can be obtained only if lactic acid bacteria and yeast present in the sourdough are brought to a physiologically inactivated state. That's why it is important to obtain sourdough, dry sourdough, in which the microflora is inactivated but still remains active after a re-fermentation and gives us a good technological parameters. So to methods used during the research I would like to present to you included 16S RNA sequencing followed by PCR reaction, and the basic local alignment search tool was also used. It allowed us to isolate and identify and after select a few lactic acid bacteria strains used for further production of sourdough and drying of that product. Another analysis conducted were total titrable acidity, volatile acidity, microbial count was performed with microbial culture or on amorous medium, and drying of the sourdough was done in a convection drying oven. What was performed, sourdough was spread evenly on stainless steel trays and then dried in different conditions analyzed during this investigation. So from artisan bakery, which uses spontaneous fermentation for their sourdough production, we obtained two types of sourdough samples, rye and wheat sourdough. After isolation and identification of many strains of different microflora, we selected four the most numerous lactic acid bacteria strains and chosen them for further analysis. For wheat sourdough, 3 strains of Lactobacillus were chosen: Lactobacillus brevis, plantarum, and paralimentarius, while in rye sourdough there was 1 additional Lactobacillus rosea. Next, prior to the drying process, it was important to choose proper starter culture, proper inoculum for our sourdough. We took 2 approaches. We used monoculture as one type of the inoculum. Monoculture meaning that biomass of each separately cultivated strain of lactic acid bacteria was added into the recipe of the sourdough and then fermented, while co-culture, 3 or 4 strains, depending on the type of the sourdough, were cultivated together, and after reaching sufficient biomass amount, they were added as a starter culture. As you can see, the microbial count of 2 sourdoughs present itself at almost the same level, giving us around 3 times 10 to the power of 9 colony forming units per gram. What was checked next, it was total and volatile acidity, very important parameters for sourdough. And what is shown, regardless of the type of the inoculum, these parameters give almost the same results. Next, again, the microbial count was performed in our different type of sourdough, but this time with intention to shorten the time of fermentation. We started with 18 hours of fermentation, and the longest time was 24 hours. So in different phase of growth of our lactic bacteria, we obtained similar results. The magnitude of 10 to the power of 9 was obtained in each of the case. That's why 18 hours was chosen as the proper duration time, optimal duration time of the fermentation of our sourdough for further analysis. As it comes to the choice between monoculture and co-culture, co-culture was chosen as it resembles conditions of spontaneous fermentation that is applied in the artisan bakery that provided us with samples. Okay, and finally, we've come to the drying process of our sourdough. 3 thickness layers, 3 thicknesses layer of sourdough were applied, 3, 5, and 7 millimeters, as well as different temperatures and time of drying were applied. Starting conditions were 45 Celsius degrees and 8 hours, which gave us moisture content at level between 12 and 22%. In order to lower this moisture content even more, the time of the drying was prolonged up to 36 hours, which gave us moisture content at around level of 5. But 36 hours is a very long time. That's why we tried to we tried higher temperature for this process. When 50 Celsius degrees were applied, we obtained moisture content at the level of around 30%. The same happened with 55 Celsius degrees. Unfortunately, with a further increase of the temperature, the moisture content didn't lower. What happened was this high temperature caused rapid evaporation of the water from the top layer, from the surface of our sourdough, creating a kind of crust which prevent the water from the deeper levels to evaporate. That's why the moisture content was still so high, even though high temperature was applied. That's why for further analysis, 50 and 55 Celsius degrees was taken. Again, physicochemical parameters of these sourdoughs were analyzed, investigated. When it comes to the total acidity, rye sourdough showed total acidity at the level of around 7 milliliters of 0.1 molar sodium hydroxide, where wheat sourdough was slightly more acidic. When it comes to moisture content in these two temperatures, rye sourdough was dried up to 10% of moisture content, while wheat sourdough obtained even lower moisture content at around 4–6%. These numbers give us good technological parameters of our sourdough, enabling proper handling and storage, and as well should give us good survivability of microorganisms present in that dried sourdough. That's why, again, microbial count was performed on this time dried sourdough, and effect of these two different temperatures and 2 different times of drying was checked. What we can see is that in case of wheat sourdough, the count of lactic acid bacteria is at the magnitude of 10 to the power of 3, regardless of the conditions of drying, while rye sourdough gives us a microbial count to the magnitude of 10 to the power of 7, but higher temperature decreased this microbial count by 100. This is a good, promising result when compared to the literature data about spray drying method, where the numbers given show that the magnitude of lactic acid bacteria that survived after spray drying is only 10 to the power of 4. So after this analysis, it was agreed that the optimal conditions for drying of the sourdough to preserve good condition of lactic acid bacteria is 50 Celsius degrees and 8 hours. And the last step of the research was to investigate how much of our dried sourdough should be used for either direct methods for baking or for activating sourdough and re-fermenting it to get a fresh sourdough. What we've obtained is that significantly higher amount of our dried sourdough would give better results and higher microbial counts, sufficient to use it for baking of sourdough bread. So final conditions for fermentation, drying, and possible re-fermentation of wheat and rye sourdough samples are the thickness of the layer should be 3 millimeters. As for the inoculum, co-culture should be used. Fermentation time 18 hours at 30 Celsius degrees, and for drying conditions 50 Celsius degrees and 8 hours. And if the sourdough is used further for baking or re-fermentation, then optimal dosage would be 50% and re-fermentation time 18 hours. To sum up, the sourdough that was dried in our institute showed good technological parameters, volatile acidity, total acidity, as well as when compared to the spray drying method, we obtained higher survivability of lactic acid bacteria. So it could serve as inexpensive sourdough starter culture, or it could be a possible alternative for popular drying methods, as well as it could serve as a possible component in a direct method of sourdough bread production. That was all from me. Thank you for your attention. If you have any questions, I'm open, and I hope that if you have an opportunity, you can visit us at the Faculty of Biotechnology and Food Sciences in Łódź.
Thank you very much, Agnieszka, for very interesting report and invitation. Any questions for speaker?
I have one question. Agnieszka, what do you think, which drying method is the more gentle and promotes the survival of lactic acid bacteria?
We designed this method to be more gentle because we use lower temperatures instead of higher temperatures used in the popular methods. For example, in spray drying, we use a really hot air to create that powdered sourdough.
Okay, thank you very much. Thank you for your invitation.
Thank you. If no one has questions, okay. Now we have finished our section. Thank you all for participation and such an interesting research. And we will be happy to see your feedback at our social media.
Thank you.
Шановні колеги, дозвольте ще раз подякувати всім, хто взяв участь у нашій першій конференції, яку організована кафедрою біотехнології Національного фармацевтичного університету. Ми розглянули дуже широкий спектр питань застосування біотехнологій у різних галузях, тому я сподіваюся, що цей захід був цікавим, ви всі отримали задоволення від цього процесу. Також сподіваюся на ваші відгуки і на нові контакти з подальшою співпрацею. Тому, якщо є якісь бажання щось сказати, побажати, може колегам, може організаторам, якісь побажання, будь ласка. Якщо ні, тоді ми завершимо нашу конференцію. Ви можете отримати сертифікати незабаром, я думаю, завтра або післязавтра на сайті нашої кафедри буде посилання на та буде можливість скачати сертифікати, а також збірник матеріалів конференції буде через декілька днів, також буде у доступі на сайті нашої кафедри. Тому все, дуже вам вдячні та до зустрічі.
Дуже дякуємо організаторам, дуже цікавий була конференція, дуже цікаві доклади, дуже дякуємо.
Дякуємо, Наталія Сергіївна.
Єдную побажань наступних Вже чекаю наступну конференцію.
Да, будемо готуватися більш ретельно готуватися до вашої конференції. Будемо дуже раді, якщо буде запрошення.
Звичайно, звичайно. Дякуємо всім вам за зворотній зв'язок. Ми бачимо у чатах також є ваші відгуки. Дуже вам всім вдячні. Вам будьте здорові. Будьте
Наталія
здорові
Ігорівна,
в першу чергу.
Дякуємо вашій
Дякую.
кафедрі за організацію, в першу чергу вам, ну і всім співробітникам, які так добре попрацювали над організацією, над проведенням цього заходу. Дуже приємно було почути дуже змістовні доповіді. Дуже приємно було побачити географію доповідей. У нас були сьогодні закордонні доповіді, що не так часто у нас буває. Це може заслуга колективу кафедри та в першу чергу завідувача кафедри. Дякуємо.
Дякую, Олександре Сергійовичу, дякую. Тому всім бажаю успіхів та творчої наснаги. Дякуємо, все, ми завершуємо. Дякую, до побачення.
До побачення.